Hi everyone and welcome to today's episode of ZK Studies, where we are going to be delving deeper into food tests. Now this is a topic in FB and we're going to see the different food groups and the reagents required to test for their presence. So let's get started. First, we're going to do a starch test. Now for starch, the reagent we need is iodine. If starch is not present, we say it's a negative test. The iodine will be orange-brown. If starch is present and positive in the sample, it will turn blue-black. Okay, now for reducing sugars, for example glucose, the reagent we use is Benedict's. A negative test result for a reducing sugar would mean the Benedict's is blue. And then there are various degrees of indicating the presence of a reducing sugar based on the sugar content and concentration of the sample. So it would turn from blue to perhaps green, yellow, orange, and finally brick red. And for Benedict's, what's really important is that you need to heat the solution. So you might use a water bath to do this. Okay, so we've done starch and reducing sugars. How about a protein test? Now to test for the presence of proteins, the reagent we're going to use is Biuret. A negative test result would mean blue, and for a positive test result, the color we're going to observe is either going to be pink or lilac. And I believe this is due to the chain length of the polypeptides. Pink or lilac will be observed. So that's our protein test. Next, for lipids. Now for lipids, there are two tests that we can do. First, we have an emulsion test. So in the emulsion test, we combine the lipid with some water and ethanol. And if lipids are present, a cloudy white emulsion will form. Secondly, we could also rub the sample of lipid onto a piece of paper, and then what happens is that if the lipid is present in the sample, the paper will turn from opaque to translucent. Translucent. So it's very slightly see-through. So there you have it, the tests for starch, reducing sugars, proteins, and lipids.